Hello everyone, I am Dr. Devi, Associate Professor, Department of Genetic Engineering, SRM Institute of Science and Technology. I am here today to explain and demonstrate a very basic but important experiment of animal cell culture, which is part of the Animal Biotechnology Practical, preparation of media. Animal cell culture refers to in vitro culture in an artificial environment in the laboratory, providing favorable conditions for the cells to grow. Animal cell culture is an important biological research tool because it plays an important role in cancer research, gene therapy, stem cell biology, and also in vaccine production. Animal cell cultures are typically grown in a liquid culture medium which contains number of nutrients, and they are cultured in culture vessels such as petri plates or tea flask. There are two types of animal cell cultures: adherent cultures and suspension cultures. Adherent cultures are those that grow attached to the substratum of the culture vessels in which they are growing, and the suspension cultures are non-adherent cultures. The culture medium contains many nutrients such as inorganic salts, glucose, vitamins, amino acids, serum, and trace elements. There are different types of animal cell culture media, but the ones which are commonly used are DMEM or Dalbecko's modified Eagles medium and minimum essential medium or MEM for adherent cell cultures, and RPMI 1640 or Roswell Park Memorial Institute medium for suspension cultures. These media are commercially available and can be purchased, or there are lyophilized powders which are available which can be used for media preparation. The other component of the media is serum, which is generally used for the growth of the cells as it contains a lot of growth factors which aid in the proliferation of the cells. The serum which is generally used is fetal bovine serum. About 5 to 20% of serum is used depending upon the cell type which you are culturing. Few mammalian cell types can be grown in serum-free media, but they have to be supplemented with trace minerals, specific protein growth factors, and other components. Antibiotics is another essential component of media, and usually penicillin and streptomycin is used to prevent the growth of bacteria and fungi. pH is very important in cell culture, and cells grow well at physiological pH of 7.2 to 7.4, 37 degrees centigrade temperature, and 5% carbon dioxide. Phenol red is the indicator which gives the pink color to the media, and if the media turns acidic, then you can see that the media color changes from pink to yellowish in color, and if it becomes alkaline, it becomes purplish in color. This is an indication that the media has to be changed. In our experiment today, we are going to use powdered media which is commercially available, and to this media we may have to add sodium bicarbonate. Sodium bicarbonate is important because it plays a critical role in maintaining or regulating the pH in which the cells are growing and acts as a buffer. Sodium bicarbonate is also a source of carbon dioxide which is essential for cell metabolism and for maintaining pH. Now we are going to demonstrate the step-by-step process of media preparation. We are going to prepare minimum essential media, and the powder has been commercially purchased. First of all, the biosafety cabinet has to be sterilized by UV for about 45 minutes before use. The person who is performing the experiment should make sure that he is wearing personal protective equipments. We should always make sure that we use sterile double distilled water. For the preparation of 1 liter of MEM media, we have to use 9.6 grams of commercially available MEM powder, and we have to add this to the sterile double distilled water and make sure it is mixed thoroughly and the powder is completely dissolved in the water. After this, 2.6 grams of sodium bicarbonate is added to the solution and it is also mixed thoroughly. Now pH has to be checked using a pH meter, and we have to make sure that the pH is within the range of 7.2 to 7.4. If there is any variation in the pH has to be adjusted. After checking the pH of the media was filter sterilized using millipore filter. Five percent of fetal bovine serum is added to the solution and mixed thoroughly. After this, antibiotics is also added to the media and mixed thoroughly. Some of the precautions that needs to be taken while preparing media is that the commercial media which you are using may be less expensive, but it has to be sterilized properly. It is always better to sterilize the media before adding serum, as addition of serum can cause foaming and it can lead to denaturation of some proteins. It is always better to add serum after the filtration. After the preparation of the media, it is always better to check to see if there is any contamination by transferring a small volume of the media onto a Petri plate and keeping it in the incubator at 37 degrees centigrade, 5% carbon dioxide for about 72 hours to ensure that it is contamination free.